Hey, thank you so much for the introduction, and I'm excited to have this opportunity. And thank you also for giving me this opportunity to share with you some of the recent work from the lab. So this is story um, originally based on our interest in understanding how the endoplasmic reticulum maintain a redox homeostasis, and we ended up discovering a novel conserved protein using a combination of computational genomic screen and a functional screen in C. elegans. So nearly all eukaryotic organisms, including human, it have uh, the organelle in the endoplasmic reticulum, and that's actually a crucial organ for a lot of function. One of the key function for ER is to enable the so-called oxidative protein folding. So this is essentially a process in which most of the secreted and transmembrane protein are imported into the ER lumen and have to undergo a normal folding process, and that require disulfide bonding inside of the protein. And this process actually involved electron transfer. So most of you are probably very familiar with the electron transport chain in the mitochondria. There's actually a similar electron transport chain in the lumen of the ER. So what happens is that most protein, almost like 40% of the protein, they have to go to the ER and to undergo this kind of folding process. They come into the lumen of the ER, and the intermolecular cysteine group, they form the disulfide bond, and in that process, they basically they donate an electron to the downstream electron acceptor, and through the chain of electron transfer that is catalyzed by a complex including PDI, protein disulfide isomerase, and ARO1, which is oxidase. And these two proteins, they use oxygen as an electron acceptor to take the electron from the cysteine group so that the disulfide bond can form inside of the protein to stabilize the folding. And the vast majority of the electron actually go to oxygen, and that leads to generation a huge amount of H2O2, because for every disulfide bond to form, there's one molecule of H2O2 generated in the lumen. And there's also a minor fraction of electron going to ERDJ5 to facilitate ER-associated degradation. But the vast majority of the electron from disulfide bond formation go to oxygen, forming a tremendous amount of H2O2. And it's estimated that there's almost 700 nanomolar H2O2 in the lumen of the ER. And this is in contrast to the concentration of H2O2 in the cytosol. So it's only about 2.2 nanomolar because
Dengke, can I ask you a question about that?
yeah.
I guess one, is there a superoxide dismutase in the ER, or is that thought to be non-enzymatic? And under, I guess, low oxygen conditions, are there additional electron acceptors that become physiologically relevant?
Right. So that's a great question. So the conventional thinking is that there's no superoxide dismutase in the lumen of the ER. It's only present in the cytosol and responsible for the conversion of superoxide into H2O2. And so that's actually the point I'm trying to make. And so there's no well-established antioxidant system in the ER and ER-associated membrane. But nonetheless, H2O2 and its associated its derivative like hydroxy radical after Fenton reaction are extremely damaging. They can cause protein peroxidation, lipid peroxidation, and oxidative DNA damage. So now the question is how so we have a variety of mechanisms to detoxify H2O2 in the cytosol, but what happens to the ER membrane and where a lot of crucial biological processes happen, like ribosomal uh, protein translation and a lot of oxygen-dependent reaction that happen there. So in the literature, there has been a lot of discussion a potential mechanism on the ER membrane responsible for detoxification of H2O2, but the molecular identity of the antioxidant protein on the ER membrane has never been determined. So we got very much interested into the problem and decided to look for such a protein. And so that is basically the initial question we wanted to address. And you may um, so it turns out when we started, yeah, I'm sorry, is there any question? Okay, so it turned out that we could learn a lot about this H2O2 detoxification from microbiologists. So decades of work from bacteria actually suggested that the mechanism responsible for H2O2 detoxification in bacteria actually are carried out by um, the transmembrane protein called SCSB and DSBD. So these two proteins, they are located at the periplasma membrane. And so the bacteria, they face the same problem. They have to fold a lot of
Excuse me, sir.
protein
I think you've been told.
outside of the cell. And so they also need to detoxify H2O2 after the oxidative protein folding and in the periplasm. And so these two proteins were identified based on their activity to reduce H2O2 into water, and also they can act on the substrate, the protein substrate um, with disulfide bond and also with peroxide uh, um, group. And so basically what happened is that they uh, use electron from thioredoxin and then transfer the electron across the periplasma membrane, and that leads to reduction of H2O2 and it's a protein substrate outside of the bacterial cell. And so a crucial feature of the function is the electron transfer mediated by the cysteine group along the chain of the electron transport route. And so this is um, it's well established biochemically. And but the problem is that there's no eukaryotic homolog that carry out a similar function that never has ever been identified. In fact, when you try to, of course, this is typically the first thing we would do to try to look for some eukaryotic homolog. But obviously, um, this this kind of the simple blast P search wouldn't work. Otherwise, we would have discovered the protein already after so many years of work from bacteria. So, but we did that kind of simple blast P search anyway, and this is basically a list of protein you can find from eukaryotic organism, um, and with a similarity to bacterial homologue DSCB and the SCSB, and there's no and it there are no obvious or apparent eukaryotic homologue identified. Some of the putative hits turn out to be bacteria contaminants, and so with now we um, have this problem. Uh, uh, we have this bacteria protein well established to carry out this electron transfer to reduce H2O2 and protein substrate uh, in bacteria. Um, and mammalian cell, including actually all of the animal cells, should have such a putative mechanism and membrane protein on the ER membrane. But the identity of this membrane protein have never been established. And so we thought that maybe this protein um, may not may actually have a structure or functional feature rather than sequence homology. You can find in bacterial counterpart. And so the functional features, I mean, the way this protein carry out the electron transfer and reduce um, the target protein or molecule. And so, based on the biochemistry of the DSDB protein, we basically read every a single paper about a DSBD protein in the literature, almost published almost 30, 40 years ago, and they um, uh, show that there are several crucial features of this kind of protein. And so the protein are the protein family members are at least four multi-pass transmembrane protein, and so they carry out electron transfer through closely spaced system pair across the membrane. And also, the transmembrane electron transfer have to occur through a nearby transmembrane segment within 10 angstrom. And of course, since we are looking for eukaryotic counterpart, we would anticipate the candidate would be broadly conserved in multicellular organism or in all animal. So when we thought about the way different ways to achieve that goal, we have we because our lab actually has been using C. elegans as a powerful tool to discover new novel conserved protein, and we thought about a potential functional screen based on the activation of the oxidative stress response. And so you can imagine that you can use the reporter to look for mutant. If a mutation if the mutation of the target would lead to H2O2 elevation and that lead to a reporter activation. But the problem is that there are basically too many ways to activate the oxidative stress response, right? So if you mess up a mitochondria, to mess up the NADH oxidase, or even to mess up to disrupt the heme trafficking in C. elegans, you have a strong activation of the oxidative stress reporter. And so we decided to basically combine a, a functional screen with a computational screen uh, using um, the um, the structure and the functional features I just mentioned to you for the bacteria counterpart DSBD, and also taking advantage of the AlphaFold predicted structure for the entire C. elegans protein. And so the way to do it is actually quite straightforward, but we are not really the expert on this, so we decided to collaborate with Tom Goddard at UCSF, and Tom was actually one of the team members responsible for the development of the UCSF Chimeric X. And so when we reached out to Tom, and he was super interested in this project, and he literally he wrote the code overnight and tested the pipeline for the C. elegans protein, and returned a list of the candidate protein in just a few days. And then we basically did the functional screen on top of that. And so the uh, the 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 flow of the computational screen is basically just like what I said. We search. We used a number of function and structure criteria to search the AlphaFold database using Camera X, and we define the criteria uh, including the proteins with close system pairs within 10 angstrom in multipass ER transmembrane segment. And we came up with a list of candidates so we can perform functional screen and validation experiment in C. elegans. So this is basically a snapshot of what we got initially from the computational screen. And so we started with 19,000, more than 19,000 uniprop protein entries with annotated transmembrane region. We ended up with more than 5,000 protein. And if we restrict the criteria for the protein with four or more transmembrane and helix structure, we got more than 3,000 candidates. And if you apply another criteria with um, at least two cysteine pairs with cysteine CXC or CXXC um, in the uh, uh, in the pool, you substantially reduce the number to 190 protein candidate. And by the way, this is CC, CXC, CXXC are based on the electron transfer pair system pair from SCSB and DSBD in bacteria. And so finally, if you pair the only focus on the protein with the paired cysteine in two helix closer than 10 angstrom, you ended up with the final 53 entries. And it turned out that a lot of them actually are GPCR among the final pool, but the 53 candidates are really manageable for us to perform a detailed functional and validation screen in C. elegans. Using, for example, the oxidative stress reporter, GST4 GAP, and H2O2 specific sensor, ORP1 low GAP reporter in C. elegans. And so we did the RNI experiment for all of the candidates, and the vast majority of them had no phenotype, and except one. This one, not surprisingly, it's non-GPCR, or maybe surprisingly, we don't actually know if GPCR would have antioxidant function to reduce H2O2. But the nice thing about this kind of screen is that we are kind of agnostic about the identity of the protein, and we did the screen in comprehensive manner for all of the candidates identified from the computational genomics. And so from the functional screen, we identified one and just one candidate that um, the loss of function of which led to a strong activation of the oxidative stress reporter. And we used two different RNI and saw exactly the same phenotype, the elevation of a baseline, the elevation of the GST4 GAP reporter. And we also confirm the causality, the specificity of the loss of function phenotype by confirming the same elevation of a reporter in the genetic knockout mutant. And so this protein is actually super interesting. When we identified this candidate, we noticed immediately that this is actually this was uncharacterized protein with the sequence name Y87G2A.13. So that simply means it has never been studied in C. elegans. And so I will talk about its conservation in just a few minutes. But before that, we want to confirm the phenotype. So as mentioned earlier, we use two different RNI and also a genetic knockout CRISPR deletion allele to confirm the upregulation of a GST4 GAP reporter. And GST4 was previously shown to require a transcription factor NHR49 and a SKIN1, C. elegans homolog over NRF2, the master regulator of antioxidant response in mammalian cell. And so we show that the knockdown or the deletion of these two transcription factors can abolish the reporter upregulation. And also pre-treatment of the reporter animal with the antioxidant N-acetylcysteine can also dose-dependently suppress the upregulation of the reporter by the RNI. Now the question, this is really because of H2O2 or something else, right? I mentioned earlier, this could be the reporter is simply a reflection of the oxidative stress response. So we validated the specificity of the response using a H2O2 sensor. So so this sensor was previously established in terms of specificity for H2O2 mediated oxidation of the sensor. And so this sensor is also ratiometric. So you basically quantify the fluorescence at two different wavelengths to know if H2O2 is elevated or not in live animal. So this is a very nice sensor. We can actually use reporter strain to quantify the H2O2 level in live animal under the control and knockdown condition. And as you can see here, in three different body regions, we see elevation of H2O2, elevated H2O2 signal, or the oxidation reduction ratio going up upon the knockdown of the gene Y87G2A.13. And interestingly, the tail region shows a very much more dramatic response than the head and the middle region, which was initially puzzling to us, but it turned out that this is simply because of the protein abundance difference in different body region. And so why do we know that? Because we generated a protein translational reporter to monitor the abundance of the protein in different parts of the animal. And it turned out that the anterior and the posterior intestines of the animal are highly enriched with this protein. And so in a zoom in view, you can see the anterior intestine, just in the anterior intestine, the GLP signal is very strong and has this characteristic structure for endoplasmic reticulum, and also similarly for posterior intestine, you see this reticulum structure. And this protein is also enriched in body wall muscle as well as sperm theca. And so this may be important for reproductive function. We'll have a piece of evidence for that a bit later. And so with zoom in, we want to confirm basically the localization of the protein and whether it's indeed on the ER membrane. So we crossed this reporter strain with a well-established ER membrane marker, and we found that they colocalize really well, suggesting this protein can indeed localize to ER membrane. And interestingly, uh, uh, when we plot the protein from this translational reporter under normal and reducing condition, we saw this redox dependent potentially oligomerization of the protein from C. elegans. Now the next question is so what, right? So this is a protein we know it's important for H2O2 um, detoxification, but what about the organismal phenotype? And so we did a comprehensive profiling of the organismal phenotype, including the animal response to hypoxia, to oxidative stress, and even under normal condition, just look at the lifespan and health span of the animal. So it turned out that the overall lifespan of the mutant animal or the um, another splicing mutant, non-mutant, and splicing mutant animal, they are largely normal. They are a little bit short-lived than white type, but otherwise is largely normal in terms of lifespan under normal condition. And we also tested the oxidative stress condition, and they are comparable to white type as well. And so we confirm also under the knockdown of the skin one condition. Skin one is an NIF2 homolog. When we abolished the antioxidant and the transcriptional response, we'll still see comparable response on phenotype in white type in the mutant compared to white type. And the only organismal phenotype we saw was the striking reduction of the blue sites, the number of progeny generated for each hermaphrodite. In the white type, it's about 320, but this is almost halved in the mutant animal. And also in terms of behavior, we found that the animal actually showed a very weird behavior response to H2O2. And so in the white type, they show this characteristic robust slowing response. But in the mutant, they have a lower locomotion rate in the first place and a slightly increased locomotion speed upon H2O2 level, perhaps reflecting a adaptation to a constitutive oxidative stress response in this mutant. So now, before I tell you about the evolutionary conservation and heme binding and some chemical bio property of the protein, I'd like to quickly revisit the mechanism of the electron transfer. So I mentioned earlier, the bacterial counterpart DSBD use a system pair to facilitate electron transfer across the membrane to reduce the target. And so we went back to check the system pair identified by the computational genomics, and we found there are actually a few different system pairs along the transmembrane segment of this protein. And this protein we tentatively named it as ERGO1, ER protein important for redox function. And so this is a protein that also that appear to also use the system pair to facilitate the electron transfer. And obviously, that was just our hypothesis. And to experimentally verify that, we generated a CRISPR knock-in allele, converting the system pair into alanine. And we found that the alanine mutations can abolish the function of the protein in terms of activation of the GST4 GAP upregulation, and based on the transgenic rescue experiment. And as you can see, the knockout without array showed upregulation, and the with the array, one type of array, you can see restored response, restored regulation, but with the mutant array, they still stay very high with constitutive GST4 GAP expression. And so the in terms of evolutionary conservation, we found that this is actually a protein that has not been extensively studied at all in the literature. And C. elegans homolog had never been studied before. And so if you search for homolog in other organisms, you can see the mammalian counterpart named TMEM161. There are two paralogs, A and B, in most of the mammalian genome, including human. And in Drosophila, there's one single homolog, just like in C. elegans. So this is based on the sequence conservation. This is a broadly conserved protein family with understudied studied function, and there are a couple of genetic studies published very recently, just a few years ago, describing the phenotype for the loss of function of the mammalian homolog in mice and a human patient. In the mouse knockout, which is perinatal lethal, you actually see the mouse embryo with this very striking cardiac arrhythmia phenotype. And so most of the mutant homozygous, they die immediately after birth, and a few survivors are able to persist just a few weeks after birth, but they show very striking the cardiac arrhythmia, eventually leading to death of the homozygote. And in human patients, so this was on the right um, is a result from human genetic studies. They identified from a human pedigree with well, based on the MRI imaging, a dramatic phenotype of so-called brain polymicrogyria. And they traced the family tree and used exome sequencing and identified a mutation in the human counterpart in the human homologue of the C. elegans ergo1 on TMEM161B, and the mutation happens at a highly evolutionarily conserved residue of the protein. And at this point, we know this protein family is clearly very important. And mechanistically, there was no mention of how how the mutation of this, how the normal function of the protein actually maintain the cell function, and how the mutation of the two mammalian homologue led to disease in those two studies. And because this is a highly conserved protein family, so we went back to check the cysteine pairs of all the homologs we can identify. And so it turned out that most of the cysteine pairs you can find along the transmembrane segment and are also just like what we see in C. elegans, they are closely spaced to each other and potentially fulfilling a function to facilitate the electron transfer. But there are also very interesting differences. For example, in the human and in the mammalian counterpart, the cysteine pairs are enriched in the upper part, and the lower part may the electron transfer across the transmembrane segment in the lower part may be mediated by some other cofactors. So that's what I'm going to tell you actually in the next 5 to 10 minutes. So this is this was initially very puzzling to us, and we didn't understand why, but we used AlphaFold prediction and to identify a potential binding pocket in the mammalian counterpart for the heme molecule. And so but before we characterize the human protein biochemically, we wanted to confirm genetically the human homologue and the Drosophila homologue have a similar function using a rescue assay in C. elegans. And indeed, you can see the transgenic expression of Drosophila, C. elegans, Drosophila, and human homologue are able to suppress the upregulation of GST4 GAP expression in the C. elegans non-mutant, and indicating the function of this protein family is broadly conserved. And also human homologue 161B is also localized to the ER based on the colocalization with a well-established ER membrane marker. So now I've told you about the conservation of this protein family and the function of this the C. elegans homologue in H2O2 detoxification and its mechanism in its potential mechanism of electron transfer through the cysteine pair. But one puzzle for us was how this human protein actually it could potentially mediate the electron transfer since the cysteine pairs are all enriched in the cytosolic side. And so we by the time we discovered the C. elegans homologue, the AlphaFold 3 came out, and that tool basically allowed you to assess the binding of your target of interest to a variety of ligand, including, for example, NAD, NADPH, and FMN, and heme molecule. So we did that kind of screen, not frankly not a comprehensive screen since the pool of small molecule ligands is quite limited. But nonetheless, we were able to find the protein, the human ERGO1 homolog 161B can is predicted to bind to the heme molecule. And also from the structural point of view, you can see the AlphaFold 3 algorithm predicts the coordination of the heme molecule by this tyrosine residue, which is also highly conserved from C. elegans to human. And so we also purified this protein from HEC293 cells, and based on the absorption, we noticed that this mutation, the tyrosine, the white type protein show a characteristic absorption band for heme, and the tyrosine mutation can abolish the absorption spectra of the protein, and consistent with the notion that this protein may indeed be able to bind to heme molecule. And functionally, we also showed that this the tyrosine is crucially important for maintaining the function, maintaining the function of this protein in rescuing the C. elegans null mutant. Now, just very quickly to introduce the heme biology. I'm sure most of you are quite familiar with this literature. So the heme is a crucial biomolecule for life. And so in most of the in most organism, heme is biosynthesized in mitochondria and is exported into the cytosol in two different pools, labile pool and a immobile pool, and they are delivered to the target protein, or they can function as a signaling molecule, and or even act as a ligand to activate transcription to maintain heme and redox homeostasis. And the C. elegans is kind of strange. It doesn't synthesize the heme molecule. It doesn't even have the enzyme for heme biosynthesis or the heme oxygenase. But nonetheless, C. elegans has all of the proteins for heme absorption, uptake, and trafficking, because perhaps because C. elegans takes the heme directly from its food, bacteria, and they need all the machinery to manage heme nonetheless. And so one crucial heme trafficking regulator is the protein, the well-known protein GAPDH. So this is a protein canonically known to be important for glycolysis, but has moonlighting function as a heme chaperone to help the trafficking of heme throughout of the cell to the target. And so this was largely done in the mammalian cell, and we wanted to basically replicate the binding. Wanted to test whether indeed GAPDH can function as a heme chaperone, and more importantly, whether the human homolog can bind to GAPDH directly. And so this is in collaboration with Zhiyong Low's lab in Tsinghua University. And so they basically purified both TMEM and the GAPDH protein directly from the HEC293 cell, and the and then they used both protein to do the co-fractionation and spectral analysis, the absorption analysis, and so this data suggests that they are able to co-migrate after the purification, these two protein, and also the spectral analysis indicate that the human homolog TM161B can form both monomer and a polymer, and this absorption for heme indicate that the polymer, the oligomerized form of TMA160B appears to have higher affinity for heme molecule. And so this is quite preliminary, so I don't want to stress too much about the interpretation. So this we are still working on this protein biochemically, and also because this protein is a transmembrane protein, so we rely on this collaboration with the structural biologists to try to resolve the three-dimensional details of the protein and its interaction with the GAPDH. And so, I just also want to mention that a very recent paper published basically a comprehensive list of all the heme-binding protein in human cell using a heme-based photo affinity probe. And so in this approach, they use UV light to stabilize the conjugation of the heme-binding protein to enrich the heme-binding protein for mass spectrometry analysis. And among the hundreds of candidates, TM161A appears in the list, and this is just one of hundreds of candidate heme-binding protein, and obviously it requires confirmation, but nonetheless, it appears to be consistent with what we have seen for the human 161B protein as well, based on its heme-binding property. And the final piece of data I want to share with you is also unpublished. Is the preliminary cryo-EM analysis of the human 161 and on the heme chaperone GAPDH from the HEC293 cells using a nanodisk preparation, and our collaborator was able to obtain the cryo-EM particle and the categorization of different classes of conformation for this protein with or without GAPDH in the presence of a nanodisk conformation. So one interesting thing is that on top of this nanodisk, you can actually see the cap of the TMEM161 on that the electron density appears to be consistent with GAPDH bond heme molecule. So they are doing detailed analysis to determine whether that electron density indeed correspond to heme bond GAPDH, and to help understand how the heme molecule may actually be incorporated into 161B, the human counterpart of the C. elegans homolog we initially discovered. And this is just to show some very unexpected observation when they try to determine cryo-EM structure. from the negative stain sample, they noticed that the protein, the purified 161B, actually formed this striking filament structure under normal condition, and that can be changed into a dispersed configuration with the reducing agent, the DTT. We don't know what that means, but we can only speculate at this moment that this is a likely a way for the protein to change the affinity for heme, because the earlier I showed you the spectral analysis that the oligomerized form has a higher affinity for heme. And so this may indicate that this protein inside of the cell can change the heme affinity based on the oligomerization state, and as a way to even regulate heme labile heme pool inside of the cell, and to eventually facilitate the electron transfer across transmembrane membrane, either to reduce H2O2 or to reduce other protein target with peroxide or disulfide bond. And finally, this is a schematic of the model. We think this is a evolutionarily conserved ER membrane protein that got against peroxide stress in eukaryotic cell on the animal cells. We showed the evidence from C. elegans in the human cell. And so the protein also used the cysteine pair, just like in bacteria, but also different to bacteria, they may be able to use heme from cytosol carried by the heme chaperone to transfer the electron together with the cysteine pair to the target molecule H2O2 and other ER protein target with disulfide bond or peroxide group. And in the absence of the protein, you have elevation of H2O2 and abnormal oxidative protein folding, leading to misfolding of the protein inside of the ER lumen, activation of the HSP4-GAP, and the impaired reproduction and behavior response to oxidative stress at the organismal level. This is just a recap of what I have just told you. We use a combined computational functional approach to discover a long-third ER guardian of peroxide stress. And we showed this striking phenotype in C. elegans, and also the conservation of this protein function in the animal cell based on the rescue of the C. elegans mutant phenotype. And some of preliminary biochemical analysis indicate this protein may also bind to heme, likely involved in electron transfer. And since the loss of function of this protein in human can cause disease with unknown mechanism, we hope our studies may also indicate a potential options for the treatment. For example, if the disease phenotype is caused by oxidative stress, we might be able to alleviate that with a certain antioxidant in those patients. And with that, I'd like to thank people in my lab who did most of the work, and also our collaborators from UCSF, and also Paul. I reached out to you to ask you about this protein, and I got a lot of advice and very valuable suggestions in terms of biochemistry and its physiological function in the cell. And also our funding support from NIH and Center for Iron and Heme Disorder at Utah, University of Utah. Thank you all, and I'll be happy to take any questions. I think I still have few minutes.
Yeah, thank you so much. You do still have a few minutes. So if you have questions, please just unmute yourself and ask.
Yeah, thank you. That's great. I had fun to see the latest on there since you sent me a draft of your paper. I don't know if that was a year or two ago, but yeah, I had two questions, somewhat related. I guess the first is, how do you think about the orthology between the C. elegans and TMEM161? It seemed like your phylogenies suggested the C. elegans was in a divergent outgroup relative to the other, minimally mammalian orthologs. And it looked like the C. elegans protein may have had multiple additional transmembranes, and it wasn't clear you had evidence that it bound heme. And then I had a second question about your UV vis spectrum of the bound heme, which is almost looks like it doesn't have axial coordination. It almost looks like what you'd expect for uncoordinated heme and buffer. Tyrosine's pretty uncommon as
Mm-hmm.
a heme ligand. There are some, but it's relatively uncommon. And I was just curious if treated with a reducing agent, if you actually saw a Sorey peak or evidence for spectral changes.
Mm-hmm. Thank you. That was a really good question. So we did a rescue experiment, although we saw the partial rescue, but was not complete compared to C. elegans white type version. So indeed, as you pointed out, there might be some divergent function in C. elegans versus the human counterpart, especially whether C. elegans homolog can bind to heme or not. We don't know yet. It didn't show up in the AlphaFold 3 prediction. And we are trying to purify the C. elegans protein using GIP affinity nanobody and to do the same kind of experiment as we did for human protein. And the second question relates to the heme coordination. Yes, so that tyrosine residue is highly conserved, that's for sure. But we don't know if that's also used for heme coordination in C. elegans. And even for human counterpart, the 161B, we showed the spectral analysis is important for heme binding, but we don't know if that residue may also affect the protein on the conformation, for example, to indirectly impact the heme binding. So that requires a lot of more in-depth analysis. And just one final note about this heme binding, we do think the heme binding may not necessarily tied to the electron transfer only. As you pointed out, there's a possibility that this protein may form a heme binding pocket to temporarily buffer labile heme or even as a storage site for cytosolic heme pool.
Great, thank you.
I have a question.
Hi, yes.
Hi. So I was thinking about the implications for the biology of this protein. And as you know, not all cells in metazoans secrete proteins equally. Maybe in C. elegans, the intestinal cells are hypersecretory.
Yeah.
But in mammals, one example of a hypersecretory cell is the beta cell in the pancreas,
Mm-hmm.
which expresses huge amounts of insulin, which, as you know, is a disulfide bonded protein. It has multiple cysteine pairs.
Yeah.
And you get this oxidative stress in beta cells, which can cause beta cell dysfunction that can contribute to type 2 diabetes. So I guess my question is, in these different tissues like plasma cells and beta cells that have very high secretory load, do you see different levels of this protein? And do you think there's some potential contribution of this protein to diseases like type 2 diabetes?
Mm-hmm. Yeah, so there is a single cell RNA-seq data set out there, and also proteomics data. So this protein appears to be indeed enriched in some secretory cell, like a plasma cell. We don't know why. Maybe as you said, as you just mentioned, that they require high demand for the disulfide bond on the formation for protein that are secreted. For beta cell, we haven't really checked, and we hope that eventually someone or maybe by collaboration we will be able to generate knockout mice to test the function of this protein, mammalian protein, in like beta cells, in plasma cell, and in disease model. And then we hope to be able to use, for example, antioxidant or some protein to alleviate the oxidative stress in those disease model.
Yeah, great. Thank you. There's a big diabetes center at UCSF, so multiple people
Yeah,
you can contact there.
thank you.
Hey, Dengke, this is more of a plug maybe. So I'm assistant professor here at the University of Utah.
Hmm.
I run the protein metabolite discovery core that's part of the CIHD,
Hmm.
and it looks like you have maybe with collaboration a pretty robust protocol to purify the TMEM 161.
Hmm.
I think it'd be super fascinating to put this through the platform, the technology that we have to look for metabolite ligands, specifically for maybe regulatory interactions. So I'll follow up with an email. I just wanted to throw that plug out there and introduce myself.
Hmm. Yeah, so that'd be fascinating. We are geneticists and cell biologists, and we would really love to collaborate with biochemists and structural biologists and people with expertise in metabolite and mass spec.
Excellent. Great. Thanks.
Thank you.
Any other questions? No? Okay, great. That was fantastic, Dengke. Thank you so much.
Thank you, Shannon.
I'm so glad we formed some collaborations with the
Yeah,
CIHD
we are
during this.
actually had some. I didn't talk about that at all. We had some very interesting collaboration with James on the metabolomic analysis of the extreme hibernator cell. Yeah, maybe next time I will talk about that story.
Yeah, next year.
Great. Thank you so much.
Thank you so much.
Thank you, everyone.
Thank you,
Bye.
everyone, for coming.
Thank you.